Ya, baik. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang Bapak Ibu sekalian and good afternoon Leng and also Nicole from Azure Biosystems, dan selamat siang juga Dr. Febriana dari Universitas Indonesia. Terima kasih sudah bergabung. Thank you for joining us. And also we are very grateful for your presence and willingness as one of the speaker of today's webinar. We also have Nicole as Regional Business Manager from Azure Biosystems. Thank you, Nicole, who helped organize this event. Thank you for joining. Dan kepada Bapak Ibu sekalian, para peserta webinar, kami sampaikan terima kasih yang sebesar-besarnya sudah bergabung bersama kami dalam webinar Biosm Azure hari ini.
Selamat siang, Dr. Febriana. Good afternoon, Nicole and Leng. Nice to see all of you.
Hi, Bonita. Good to see you too.
See you.
Baik, Bapak Ibu sekalian, sebelumnya perkenalkan saya Yahya Ramadhani dari Biosm selaku produk spesialis protein teknologi yang akan bertindak sebagai moderator pada webinar Biosm Azure hari ini dengan topik Western Blotting, the Principle and Applications. Berbicara mengenai Western Blot, seperti yang sudah kita ketahui bersama bahwa Western Blot merupakan salah satu teknik analisis dalam biologi molekuler dan juga imunogenetik dalam mendeteksi suatu protein spesifik terhadap suatu sampel. Bapak Ibu sekalian, sesuai dengan topiknya, webinar ini akan membahas lebih detail mengenai prinsip, pengerjaan, teknologi terbaru, dan juga berbagai informasi aplikasi yang dapat diterapkan dalam pengerjaan Western Blot, sehingga diharapkan mampu memberikan manfaat dan wawasan yang lebih luas terhadap penggunaan ataupun metode Western Blotting. Adapun rundown dari webinar hari ini, di antaranya yang pertama akan disampaikan langsung oleh Ibu Lianita Atmaja selaku Marketing Manager di Biosm, akan menyampaikan Biosm Introduction, and the next session will be present by Dr. Leng from Azure Biosystems, with topic Western Blotting, Principle, Antibody Detection, Imaging, and Normalization. And next session, application of Western Blot in biomedical research by Dr. Febriana from Indonesia University. And the last session, we have a Q&A session. So selain itu, penting juga untuk kami paparkan Bapak Ibu sekalian mengenai rules, di mana peserta kami harapkan untuk mengikuti webinar dari awal hingga akhir. Dan di akhir sesi nanti kami harap Bapak Ibu sekalian bisa mengisi form feedback yang nantinya akan digunakan sebagai daftar hadir dan juga sebagai bahan untuk pembuatan e-sertifikat yang nantinya akan kami kirimkan ke Bapak Ibu sekalian. Baik, Bapak Ibu sekalian, langsung saja kita melangkah ke agenda pertama, yakni Biosm introduction yang akan disampaikan langsung oleh Ibu Lianita Atmaja. Kepada Ibu Nita, kami persilahkan, Bu.
Selamat siang semuanya. Terima kasih Dani atas waktu yang diberikan pada saya. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang Bapak Ibu sekalian. Salam sejahtera untuk kita semua. Terima kasih sudah hadir pada webinar Biosm Azure pada siang hari ini. Pada kesempatan siang hari ini saya akan memberikan sedikit introduksi mengenai PT Bioscience Medica Indonesia dan juga bahasan webinar pada siang hari ini. PT Bioscience Medica Indonesia dikenal dengan nama Biosm yang mempunyai moto Bringing Impact and Outcome in Science Movement. Biosm sendiri berdiri sejak tahun 2011 sebagai perusahaan yang mengarah kepada riset-riset ilmu pengetahuan dan teknologi kesehatan. Kami ingin berkontribusi di sektor kesehatan di Indonesia dan meningkatkan nilai dari inovasi kepada seluruh bangsa dan negara Indonesia. Selanjutnya, produk-produk yang ada di Biosm meliputi 4 macam teknologi, yaitu dari teknologi DNA, teknologi sel, teknologi protein, dan diagnostik serta terapi. Kami mengageni produk-produk internasional yang sangat berkualitas. Juga kami mengageni alat-alat selain daripada reagen, consumable, dan bahan-bahan kimia untuk kebutuhan laboratorium Bapak Ibu sekalian. Kami juga menyediakan after sales service yang dikerjakan oleh tim Field Application Specialist dan Field Service Engineer kami yang sudah di-training secara langsung oleh prinsipal-prinsipal kami yang dengan baik. Aktivitas dari Biosm beragam, kami sering mengadakan presentasi saat ini tentunya secara online. Kemudian kami juga bisa mengadakan demo, beberapa alat kami yang stok juga secara online. Bila dibutuhkan, kami juga akan membantu Bapak Ibu sekalian di laboratorium menggunakan prokes sesuai prosedur yang berlaku saat ini di masa pandemi. Kami juga menyediakan mobile lab, yaitu lab mobil yang bersertifikat BSL 2. Adapun mobile lab kami ada berbagai ukuran, dari yang small, kemudian yang medium dengan tipe karavan tipe 20 feet, kemudian yang tipe L sebesar bis, kemudian yang tipe SL berupa kontainer 40 feet. Demikian introduksi mengenai produk-produk yang kami bisa sediakan. Kemudian pada siang hari ini, webinar kami berjudul Western Blotting, The Principle and Application, yang ada dua pembicara pada siang hari ini. Jadi, teknologi Western Blotting ini terus berkembang untuk membantu para ilmuwan menjadi metode yang populer untuk menganalisa ekspresi protein. Kemajuan teknologi telah meningkatkan pendekatan ini, termasuk pengembangan uji kuantitatif, kemampuan untuk mendeteksi banyak protein secara bersamaan, dan penggunaan sistem penangkapan digital yang canggih untuk deteksi dan analisa yang lebih mudah. Western blot juga memainkan peranan penting dalam penelitian biomedis dan klinis, antara lain digunakan untuk mengidentifikasi protein spesifik, baik berdasarkan ukurannya maupun dengan ikatan antibodinya. WB juga menyediakan data penting dari isolasi dan pemurnian protein tertentu. Secara keseluruhan dapat dikatakan Western blot menyediakan data yang berharga untuk analisa lebih lanjut dan penerapan penelitian biomedis. Miss Leng pada siang hari ini akan menjelaskan tentang dasar teknik Western blotting dengan gambaran umum mengenai teknologi ini. Sedangkan Dr. Febriana akan memberikan sajian mengenai prosedur Western blot dan elemen-elemen kuncinya, kemudian data penelitian yang dihasilkan dari Western blot, kemudian penerapan Western blot, termasuk identifikasi dan kuantifikasi protein di bidang biomedis. Dengan demikian, Biosm Blot dapat membantu mengkonfirmasi hasil ekspresi dan pemurnian protein dan reaktivitas terhadap berbagai plasma manusia. Jadi demikian yang kami sampaikan mengenai webinar pada siang hari ini. Semoga bermanfaat untuk Bapak Ibu sekalian. Selamat mendengarkan. Terima kasih atas perhatiannya.
Baik, terima kasih Bu Nita atas penyampaiannya. Melangkah ke agenda berikutnya yakni pemaparan materi dari para narasumber. Namun sebelumnya saya akan menyampaikan speaker profile dari narasumber kita pada hari ini. Jadi Bapak Ibu sekalian, agenda berikutnya merupakan pemaparan mengenai Western Blotting Principle Antibody Detection, Imaging and Normalization yang akan disampaikan oleh Dr. Kailang Tan dari Azure Biosystems. Pembicara pertama yakni Dr. Kailang Tan, di mana Dr. Kailang Tan as Regional Field Application Scientist from Azure Biosystems. She had scientific experience in School of Biomedical and Pharmaceutical Science, Guangdong University of Technology, and also Department of Anatomy, National University of Singapore. She also had scientific appointment as scientific reviewer in neuroscience research notes and frontiers in microbiology, and also as member of Malaysian Society in Neuroscience. And she also have some international publication in 2020. So, Dr. Kailang, good afternoon. Can you hear us clearly?
Yes.
Yeah. Bapak Ibu sekalian, just for your information that Dr. Kailang will present about principle and fundamentals of Western Blot, yang tentunya akan menjadi informasi penting bagi kita semua, khususnya terhadap penerapan Western Blot. So, Dr. Kailang, do you still need more time to prepare your presentation, or can we start now?
Yeah, we're all good. So let me just take over the session and start the presentation.
Okay, everything looks good, and Dr. Kailang, time is yours.
Yeah, thank you. Thank you, Danny. Right, let me share my screen. So in the meanwhile, please let me know that you can hear my voice clearly.
Yes.
If you can hear my voice clearly, please type yes in the chat box. Just to let me know everything runs well, and you can see the slide properly. You can see my laser pointer.
Yes.
All right. Okay, great. Thank you. All right. So welcome everyone to this webinar session. This session, I will be talking about Western blotting. In general, I'll be explaining the principles of every single steps of Western blotting protocol, the part where we will detect the antibody, a few of them, the methodology, imaging, what we should be taking care of, and also the final part where we touch a little bit about normalization. I am Kailing, the Regional Field Application Scientist of Azure Biosystems. So let us look at the agenda for my session. Right, first thing, definitely we need to go through the principles of chemiluminescence Western blotting. Then we will look into antibody detection with chemiluminescence and fluorescence methods, imaging, Western blot, and finally the normalization of your Western blotting result. All right, so before we start, let me introduce Azure Biosystems to you. We are a United States-based company in California. We're a team of highly experienced scientists, engineers, and entrepreneurs dedicated to accelerating your science. Our mission is to giving you raw data that you can rely on using simple workflows that you can trust. Azure Biosystems is founded by Arnold Shifji, our CEO and chairman. He is a venture investor, and also Dr. Chair. Dr. Chair is our executive vice president of R&D. If you're looking through his past experience, you will know that he's extremely experienced in terms of developing imaging system for protein expression, such as in Illumina and Alpha Inotech. I believe that if you've been doing Western blotting, Alpha Inotech will be a brand that you will be very familiar of. The core member of Azure Biosystems are all coming from Alpha Inotech. This company is fairly new, founded in 2013. At the moment, we have a few key instruments in our company portfolio. The first thing will be the industry first hybrid CCD camera imager and also laser scanning system, where we call Sapphire Biomolecular Imager. This is our highest spec instrument so far. And then we have also an imaging system that's focusing on imaging Western blot, including chemiluminescence and fluorescent Western blot, where we call it Azure Imaging System. And also we have ventured in the genomic market last year, where we launched our flagship Azure Cielo real-time PCR system. Right, let us dive back into Western blot. Okay, so this year, the year of 2021, is the 41 years of Western blotting. This whole figure is taken from the Journal of Proteomics published last year. It shows that the proportion of 5 different protein-related techniques in terms of the publication estimated by the number of text word inside the text or the title, you can see that across these 5 techniques, Western blot, the existence of Western blot, the word in the title, has been declining across the years. The percentage has been reducing, but this technique has been consistently higher in proportion when compared to other techniques in doing the protein expression study. So it shows that Western blot is quite the essential and widely used analytical technique in protein studies. Right, Western blotting is very universal. First, when you run Western blot, you need to ask, why would you run a Western blot? Do you have certain scientific questions that you need to answer? Here we break down into 2 major categories of protein expression question. The first will be qualitative, means do you need to check there's a presence of absence of a protein of interest? Like for example, is the protein A expressed in this particular tissue or not? So this is qualitative question. Second will be something that we normally do, which is quantitative question, where we relatively compare 2 or more groups of samples to accurately measure changes in protein expression. This is the simplified flow chart of Western blotting flow. So it starts with gel electrophoresis, transfer step, okay, blocking, and then primary antibody incubation, secondary antibody incubation, ECL detection reagent. Wait, if you're doing chemiluminescence detection method, then finally imaging and analysis. I will go into each of these steps to tell you what this step is meant to do in order to get the final protein expression result. Right, the first thing, gel electrophoresis. This is the part when you load your protein sample into a gel. Here will be polyacrylamide gel, and then run it through gel electrophoresis in order to physically separating the proteins from one and another across the entire gel matrix with the electrophoresis. Here we'll be saying it's a kind of simplified summary of the whole gel electrophoresis process, where you mix protein sample with loading buffer. So your protein will be loaded inside the well, load into the gel on the well, then subjected to an electrical current which applied to this gel or buffer system. So one thing to separate the proteins, you need to use gel. So commonly we use SDS page gel. The reason being it can resolve or separate the proteins, okay, based on their molecular weight. And then also the percentage of the gel does matter here. As you see here, the higher percentage it goes, the smaller the molecular weight, it can resolve or separate it better. So there are different types of gel over here. So we get SDS page gel, we got laminate discontinuous gel, we have 2 separate, or say two gel stacking on top of each other, where you have stacking gel and resolving gel at the bottom for more defined band for that in that purpose. And then you have also the commercial gradient gel ranging from 4 to let's say 20% or 4 to 15%, allow you to resolve multiple proteins that have a different wide range of molecular weight. Right, since you're running a gel, you need buffer. Standard SDS PEG gel use alkaline buffers such as 3-glycine for mid-sized protein resolution. So this is basically the part for gel electrophoresis, just to separate the protein according to their molecular weight. Next, after you separate the protein according to their molecular weight, you need to transfer these proteins to a solid membrane support. So you can do the subsequent step, like detecting the proteins and so on and so forth. So here, there are two main membranes that you can use. First will be nitrocellulose membrane, and second will be PVDF membrane. Each of them have their own perks and also disadvantages, I'll say. Nitrocellulose in this sense is much more cost-effective than PVDF membrane. Itself, nitrocellulose has low background for chemiluminescent detection. It doesn't need activation like how you would need for PVDF membrane, but this type of membrane, nitrocellulose membrane, is rather fragile and it cannot be used in fluorescent detection method. PVDF, on the other hand, it is much more durable. It's suitable for both chemi and fluorescent detection, but it also requires activation with methanol prior to use. If you're doing fluorescent Western blot, low fluorescent PVDF membrane is the best choice in terms the choice of membrane. Right, so transfer. Here we have two main transfer systems. The first thing will be quite conventional, which is a wet transfer setup, and you have semi-dry transfer setup. Each of them have their own advantages as well, where it listed over here. For example, wet transfer, it is very flexible. You can adjust the transfer condition based on the protein of interest that you want to detect. And then because it is a wet transfer, you soak the entire transfer sandwich inside the buffer. The transfer is much more, I'll say it is longer, but it is much more complete, I'll say. And this is what we recommend you to use if you want to do quantitative Western. Quantitative Western here means you want to accurately measure the changes or the relative expression between one sample and another. We highly recommend you to do wet transfer in this sense. Of course, this wet transfer have some disadvantages. For example, the buffer, if they heat up, you can interfere with the transfer. So the transfer efficiency would not be 100%. And you would need cooling mechanism, cooling unit, or even on ice or cold room during the whole transfer, because it's a long transfer. So you will need to do it at a constant, stable temperature across the entire transfer process. And thirdly, definitely you need a lot of transfer buffer because you need to fill out the entire transfer tank. Semi-dry, on the other hand, is, let us say, semi-dry, means it doesn't really soak the entire membrane in the buffer like how you did in the wet transfer. Rather than your transfer sandwich is already pre-soaked in the transfer buffer. So the whole sandwich is wet, and then you have the two electrode squeezing them in the middle, okay, then you transfer the protein. Right, so this is semi-dry transfer. It is quick, used in high voltage, okay, but you need very little buffer, or say not very little, but you need lesser buffer, definitely lesser than wet transfer. It's easier to set up, and then it's all good because you can transfer more than one blot at the same time. But this method is not highly recommended if you are doing quantitative Western, because as I said, the transfer is quick and high voltage. The high intensity field strain will cause your low molecular weight protein to migrate past the membrane. So you will have the risk of losing the membrane because of over transferred. Okay, and then this method also quite difficult in transferring high molecular weight proteins for those that have more than 120 kilodalton. And I say this is not recommended for quantitative Western because you want to know and accurately measure the differences. So you do not want to miss or lose any protein in between. And finally, the disadvantages of this is the gel can be dry out. Right. So of course you need a buffer. The buffer here, there are several components, buffer to maintain the pH, alcohol to increase the binding of proteins, SDS detergent to promote the migration of the protein out of gel, and trypsin, and so on and so forth. So here in your transfer, you need to be careful about your transfer time and voltage. You should be optimize this setting on each transfer, especially on each or the set of protein of interest that you want to do. In general, longer transfer time with a lower current are used in wet transfer. And for semi-dry transfer, you will achieve shorter transfer time with high current. But do be careful that while proteins are generally transfer more rapidly at higher voltage, the transfer efficiency across the entire membrane or gel, the sandwich, will not be always consistent. So insufficient transfer current or time may result in incomplete transfer. So you want to transfer entirely because you load exactly the same amount, so you expect them to be the same, or else it will be hard to determine the result later on. While high current and lengthy or very long time transfer time may also result the loss of the protein because the protein will be migrate past the membrane without any retention at all. So there are a few tips and tricks on this, but do remember that you need to optimize based on your protein of interest. Right, next thing, after you got your membrane, all right, the membrane right now on the surface will have all the proteins that you loaded based on the sample, and then you want to go for membrane blocking. The blocking step will aim to remove non-specific bindings of antibodies, so you can remove all these background noises. Okay, how we do it is very simple. You just incubate it in a blocking solution. So since you incubate it in a blocking solution, you need to prepare a blocking buffer. Here, there's a buffered salt solution, either using TBS or PBS. All right, but if you're doing, let's say you want to detect alkaline phosphatase-based method, like you're using chemi with epi, or you want to detect phosphor-specific antibodies, you should use TBS instead of PBS. Detergent over here, like let's say Twin 20, is recommended to added inside the blocking buffer, so to prevent microbial growth. This is because if let's say you want to reuse the blocking buffer, then yeah, this will help to prevent the microbial growth. But do remember to make sure the detergent should be completely solubilized to prevent artifacts. And one thing to be noted for is if you're doing fluorescent detection method, you shouldn't use any detergent at all throughout the entire process. Okay, so this is Twin 20 is only applicable for chemi Western. Right, blocking agent here, we have protein type and protein-free type. Protein type, you can use non-fat dry milk, which is a skim milk, which it is very economical, cheaper, easier to prepare. Okay, but it's not suitable for phosphor-specific antibody. You can also use serum, okay, but it's less favorable because it's highly interact with antibodies later on. It will give you high background. BSA, this is very good if you're doing phosphor-specific antibody incubation later on, but do not use it on tyrosine phosphorylated protein content. And it also cannot be used in anti-lectin antibodies. So the general type is go for skim milk protein blocking agent. If it doesn't work, then you go for the other blocking agents. You can also use protein-free blocking agents, such as the one that we commonly use in Southern blot, which is the water-soluble polymer, polyvinyl pyrrolidone. Okay, this can be used in Western blot as well, right? So blocking condition, depending on your experiment type and your antibody specificity or the protein or your protein interest expression and so on. But generally, we want to block in room temperature for 1 hour, gentle agitation, right? So to make sure the whole entire blot is blocked properly, right? If you want to block for more than 2 hours, you should do the incubation in 4 degrees Celsius. Right. Next thing is primary antibody and secondary antibody incubation. In terms of antibody, we have two types of detection over here. The first thing will be direct detection. The second will be indirect detection, where you have a secondary recognizing the primary with the target complex. Okay, so the direct detection is very easy. You just need to use one antibody, which is your primary antibody. This primary antibody has been tagged with either enzyme or fluorophores or any detector molecules, so you can straight away go to imaging later on. Secondary, indirect detection, you will need to form a complex between the secondary antibody, primary antibody, and epitope. Then finally, the detector molecules over here. So indirect detection is much more popular, much more sensitive because it's amplifying the signals, but it's highly cost effective as well. Right. So in terms of primary antibody, you can choose between polyclonal or monoclonal antibody. Polyclonal means it has a mix of individual antibody that recognizes different region of the same antigen. It offers wide range of binding specification, means you can easily bind the protein of interest, but sometimes you may get more than one band that we call non-specific bindings or even higher background signal. Okay. Monoclonal antibody, on the other hand, it recognizes the same epitope on the antigen. So it gives less robust signal, but it is highly specific. You usually will only see one band if you use monoclonal antibody. But if you let's say see more than one band, then it may remain true, or you just need to check whether there is another protein that has the same epitope as your antibody. Next thing, if you're doing indirect detection, you would need to incubate with secondary antibody. Here, you need to be careful about the pairing of the species. Okay, for example, you're using rabbit antibody in primary antibody, then secondary antibody must be, let's say, something anti-rabbit secondary antibody. So this is a pairing that you need to know. The class of types of primary antibody does matter as well. So you would need to depend which heavy or light chain make up the antibody, so you do the proper combo and pairing. Antibody conjugate here will be talking about the one that give you the signal. Normally will be, if you're doing chemi, will be enzymatic labels, either alkaline phosphatase or horseradish peroxidase HRP for chemi. If you're doing fluorescent Western, then we'll be using 404, the tag will be 404. And sometimes you would need to use biotin labeled or biotilated antibodies. Okay, that one is much more, pretty much for sensitive detection for low abundance protein. Right, but today in this context, we will be talking about chemi Western. Right, so we'll talk about electrophoresis, separating the proteins according to molecular weight, transfer the protein from the gel to the membrane. So now the membrane will be having all the proteins from the sample, then you block the unnecessary background noises with the blocking buffer, then probe with primary antibody of the protein interest that you want to know, and then secondary antibody. Then next we will move into this part where it is the antibody detection with either chemiluminescence or fluorescence method. Over here. Okay, right. So there's pretty much one not only chemi and fluorescence detection method. Sometimes we also use colorimetric detection in Western blot. Okay, this is very easy. You have a secondary antibody that have an enzyme reporter that catalyze, they give you go through a chemical reaction and then give you a colored insoluble product. All right, so this color will be formed on the surface of the blot, and then so you can observe by naked eyes. Then you do not need any digital camera to capture it. You just need to either scan it or by eyes, then you determine the result. But if you want to go for quantitation, I would not really recommend this method because more often we need to have housekeeping, normalization, and so on and so forth. So this method is only for one protein, one blot at a time. And then the blot could not be really used like strip and reprobe later on. All right. All right, so next let us move on to chemiluminescence antibody detection. This is a very, very popular indirect detection method. What you have is your primary antibody recognizing the epitope of your protein of interest, secondary antibody that tag with HRP enzyme. Then after that, you use ECL substrate to enhance the chemiluminescence signal. This method, by far, we say it best give you semi-quantitative, I'll say, power, but it is super good in terms of detecting low abundance protein and one single protein at a time. Okay, and you can see this graph over here. The longer the the chemiluminescence signal goes, okay, it doesn't mean that you have the same band intensity for different abundance of proteins. Okay, so there will be high chance that your high abundance protein will be saturating the membrane or your imaging system. Here, this is an example of what you see in chemiluminescent Western detected on X-ray film. Right. So the amount of chemiluminescent signals on your Western blot is subjected to a lot of variation because this is a chemical reaction. That's why we call it chemiluminescent. Okay, and the reason being that the kinetics, that the factors that affect the kinetics will be either temperature, whether it is in the dark or not, the substrate concentration, how much epitope you have, abundance of the protein, and enzyme concentration, temperature, and so on and so forth. So here we have a few example of the ECL substrate available on the market. For example, Armasham ECL, ECL Plus, Super Signal West Dural, and also Radiance from Azure Biosystems. When you plot the chemiluminescent signals according to the protein load, increasing protein load, you should observe what we call as signal linearity. What it means is the amount of signal should be representing the actual amount of the protein itself. Okay, so that's why you sometimes, if you see that in certain proteins that you detected, it always saturated, then you need to change your ECL substrate to a lesser sensitive one because you want them to fit within this optimal signal linearity range. Okay, and all of these, I'll say some of these, you can see that when you pass certain protein load, let's say 1,000 picogram, the ECL, the chemiluminescent signals is not longer linear. Thus, we call it either oversaturated or it just not longer linear. It doesn't represent the true protein amount. And this will, I'll say, we call it fall off the signal linearity, will not longer be qualified for quantitation since it's not longer representing the true actual protein amount. You see here, it should be directly proportional. Right, let's see what's the advantages for chemiluminescent antibody detection method. This is a very sensitive detection method. It can detect up to femtogram levels of protein bands. This is of chemistry of where protocol that we're quite familiar of and quite easy to do. It is compatible with either X-ray film and digital imaging. The disadvantages for chemiluminescent to use chemiluminescent detection methods for your Western blot analysis, data analysis, is these at most can give you semi-quantitation power. Means you could not entirely or accurately measure the true, I'll say, relative expression between one and another. And then this is because the nature of this chemiluminescent reaction, because it's enzymatic kinetics. It amplifies, but it doesn't. It amplifies the signal very good, but it doesn't mean that it will give you the signal linearity. And this method at best give you one protein detection on the entire band, unless you cut the blot into separate smaller strip, so you can detect one at a time. So unfortunately, if you have more than one protein that sits similar molecular weight, for example, a total, the one that I do in my study, total step 1 protein, and another one will be phosphorylated step 1 proteins. So these two proteins are not able to separate it by electrophoresis mean because they are at exactly the same or similar molecular weight. Okay, let's say they are 72 kilodalton, another one will be 71 or 72 kilodalton. I cannot separate them, but I have to analyze them on the same plot. Then what you need to do, you have to do strip every probe. Means after you detect the protein of interest A, then you strip off the secondary antibody, wash it properly, then you go on with the primary antibody incubation of your protein interest B, and then the rest of the detection and blocking method. Okay, so this is what we call a strip and reprop process. The strip and reprop process is actually having problem because there are papers and even researchers who found out that the strip and reprop process does actually change the protein amount on the blot later on. So it's not really that convincing either. Right, so if you're doing chemiluminescent antibody detection method, you might be using X-ray film. Okay, X-ray film is super good if you're detecting very low abundance of protein. It's very good. You can let the X-ray film, the whole cassette sit there for days and months just to detect a very tiny low amount of, let's say, novel microRNA, okay, or novel protein, let's say. But in terms of the common practice that we know that the expression is quite fairly at a moderate range. It has a very limited, X-ray film itself has limited dynamic range. It easily saturated. That's why if you have a pair or a lot of protein interest, with a wide range of protein expression levels, we will recommend you to go for digital imaging. Right. So like I say, chemiluminescence itself, this method is very good in terms of answering qualitative question, means the protein is there or not, but it's pretty much not very good in answering how much protein in related to other protein or QC. QC using chemi, like checking whether your Western blotting protocol working well or not, is not really that good. Correct. So one of the perks is the protein must be well resolved electrophoretically, especially separating the controls from protein of interest. Right. Next thing, let me explain a little bit about fluorescent Western blot. Okay. Fluorescent Western blot itself is the same thing. You have primary antibody, you have secondary antibody, but instead of HRP, you have fluorophores. Fluorophores itself, they have this feature called spectral separation, means the excitation and emission wavelengths are at different wavelength. Okay, then different fluorophores will have different spectral emission and separation. That's why we call it spectral separation. And with this, you can do multiplexing, means you like how you look over here. This is just single blot. You don't need to do strip and reprop. You prop it with two or more than one, definitely more than one protein antibodies. Okay, then you do by the end of the Western blotting protocol, capture the image. That's it. That's what we call as multiplexing Western blot. Okay. Right. So fluorescent itself, it gives you much stable and linear light production. And because the only thing that changed fluorescent expression, only two things. The first thing is the power of the light source, excitation light source. The second thing will be the amount of the fluorophores. And the amount of fluorophores is will be changed based on the amount of the, let's say, the secondary antibody that tag to the primary antibody and the epitope. Okay, so in this sense, we know that fluorescent will offer more linear light production. Okay, because the light power in your imaging system will be constant. So the only thing will change the expression or the signal will be only the amount of the protein. Thus, fluorescent is much more reliable than chemi signal in this sense. Yeah, that means linear fit significantly better than others. Okay, right. So fluorescent method, the advantages for this antibody detection method is you can do multiplexing. It definitely increase your quantitative accuracy. Like I said, regardless of the amount of protein, whatever the signal you get will represent the true protein amount, because there's only two factors that change the fluorescent signals. Okay, and then one already constant in your digital imaging, so the other one will be your sample only. All right. Then fluorescent label itself is stable. You can store and re-image it later. Okay, very much like how you do your IHC fluorescent slide. You can keep it in dark and then take it out and capture later on. This fluorescent detection method, it can be less sensitive than chemiluminescent and most at picogram, okay? And you have to be careful if you're using similar reagents or membrane, because fluorescent, we want to have clean background so you can do the analysis later on. Fluorescent antibody detection method is super good especially in answering quantitative question, like how much the protein is expression is different from one another, and your Western blotting protocol is QC'd and the loading is consistent, because you will definitely see that in your blot if you load it poorly. Okay, so this is pretty much where we compare when you do chemi-Western and fluorescent Western, where we're taking into account you're detecting two proteins on the blot, normally the protein interest plus your housekeeping protein. In chemiluminescent Western, you normally takes up to 6 hours. It will take much longer if your primary antibody incubation takes overnight in 4 degrees. All right, so it takes up to, in my own practice, my chemi-Western will take up to 3 days. So Monday I do, Wednesday I get results. Okay, so that is only for like that. So that's for chemi. But if you do fluorescent, the time taken to complete a multiplex fluorescent Western will down to approximately 3 hours, right? Because you can see that it cut off. You don't need to probe another antibody anymore. Everything will be multiplexing. You're detecting more than one proteins on the same blot at the same time. Of course, the next thing is cost. All right, I know the research is also very important. We need to be cost effectiveness because it's going to do a lot of them, right? We also compare the cost. This part will be in US dollar. Between ECL, okay, which is chemiluminescent and fluorescent Western. ECL for chemiluminescent using film and CCD based imaging system. You can see that of course, the most important, the most expensive reagent in fluorescent will be secondary antibody. This is very commonly we know, but if you do fluorescent Western, it cuts off the cost in terms of the ECL reagents, the film and stripping buffer, and so on and so forth. All right, and the protein marker also you do not need to use a lot. You only need to use 10 times less the protein ladder for fluorescent Western. So all in all, in the longer run, fluorescent Western will cut down your Western blot cost a lot. Okay, right. So which is right for you? So both chemi and fluorescent Westerns, they are both excellent methods. If you can use them together to give you complementary information to enhance the insight of your scientific research. Okay, you should use chemiluminescent Western if you just want to detect a single protein. Let's say you just check whether the protein is there or not. Just either checking the antibody response, post protein purification, or the most important thing is if you have very low abundance protein, then you should go for chemiluminescent method. Fluorescent method is super good if you want to detect multiple proteins at the same time. Okay, especially those 2 or more proteins that cannot be separated by agile electrophoresis, then you need to go for multiplexing. It cuts your time and costs a lot. And of course, this will provide more QC because you lessen the steps in your Western blotting protocol. No more strip and report, continue washing and incubation. And then this is the method we will recommend to do if you want to accurately quantitate the relative expression. Okay, we will highly recommend you to go for fluorescent. It will solve. Basically, nothing would change, like I say, change the fluorescent signal except the amount of your protein. This is very reliable. Right, so this is a simplified, I'll say, flowchart or diagram to determine which is right for you. If you have if you do not have digital imager and you need to detect very, very low amount of protein, then you should go for chemiluminescence straight away. But if you have digital imager and you need to detect multiple proteins on the same blot, in this sense, perhaps protein interest A plus housekeeping, let's say, and then also perhaps another protein interest which are similar sizes, and precise quantitation, we will say that you should go for fluorescent instead. Okay, so you need to choose based on your scientific questions and whatever expression they expect to see. Right, next thing, after you got your blot, you do the detection, chemiluminescent or fluorescent. Next thing is you need to capture the image. Right, capture the image itself over here. There's a, when we see here, chemi-western, okay, we have film and X-ray and also CCD based camera. You basically what you need is you need wide dynamic range. You want to be able to capture both very low and very high signal intensity in the same image without any saturation. Okay, so of course, film itself, it has a limitation in terms of the wide dynamic range. So we highly recommend you to go for CCD-based imaging system if you need to have both very low and high, very intense span on the same plot. Right, not all CCD chips are the same. Okay, here we say that signal dynamic range and everything. CCD chip itself, they have every pixel have their own capacity, what we call as pixel well cap. Right, the size of the CCD, I'll say the size of the well cap itself will determine how many electrons you can hold in each pixel well. Right, the larger the pixel well cap, of course, you can load more electron without any saturation. So it will stay true to represent how much signal that you get to capture on the entire plot. Here we say larger well cap capacity, so more pixel can capture with light before saturation, then there's greater sensitivity and dynamic range. Fluorescent here, if you're interested, I'll just touch a little bit about this. Light source also very important. LED and laser. Generally, laser is way better than LED because it is monochromatic, higher power input, very less light leakage over here, indicated in blue, which will translate into background noises. Okay, but LED definitely more economical prices. Okay, laser is much more, a little bit more expensive, but of course the performance is super premium. Right. All right. So fluorescent itself, just a touch about this, we have two categories: visible fluorescent, RGB, red, green, blue, and near infrared, 700 and 800 channels. Okay, it's the same thing. If you do fluorescent western, you will have the fluorescent secondary antibody. Okay, right. So comparison between these two, RGB and near infrared, very easy. RGB you can do up to 3 multiplex, red, green, blue. Near infrared you can only do up to 2-plex, 2 of them. Okay, RGB itself, it has more dye choices. You have sine dyes and FGFP, RAP. And near infrared range, you have lesser dye choices. So far, at the moment, we have only have 2 dyes available at this wavelength. But RGB itself, it suffers from high background signals, okay, and also autofluoresce in the sample and membrane itself. This has been tested. You can check publication, and thus they do recommend you to go for near infrared range if you can. Right, and near infrared definitely give you higher sensitivity because of the wavelength feature itself. Right, so next thing is Western blot normalization. Hi, Danny, please let me know if the time is almost up. Right, thank you.
Okay.
Yeah. All right, so Western blot normalization. Commonly, we have 2 normalization controls. The one that we were very familiar with will be housekeeping protein, that we normally use as loading control, and then it's only one band, the housekeeping protein, right? Just one band. So the one band will be the entire control for the entire membrane. So we see that if it doesn't transfer well or lesser expression, we sometimes will see that, oh, but probably my transfer isn't correct or something, or the efficiency wasn't good. Okay, but do remember that this is only one band, all right? And we often use highly expressed protein, for example, GAPDH, actin, and some others proteins that to use as our housekeeping proteins, right? And here I want to address certain issue about housekeeping proteins. Some of you might know this already, or maybe notice in your own research. Okay, so the reason for us to use housekeeping protein as a normalization control, because firstly, we trust that it is constant. It should be constant because normalization should be constant, right? It has stable expression. That's what we expect it to do. And we do expect it to allow us to QC your entire Western blotting protocol, like loading, running gel, or even transfer. Okay, but in reality, you will notice that even similar experiment, different time point, harvesting time point, tissue type, even different doses of treatment, DC state here stated it, cell tissue confluence, cell confluency as well. They do change these housekeeping proteins expression. They do change, okay? But if you have do proper categorization on your housekeeping protein, like throughout the loading of your entire protein samples and then increasing amount that it shows signal linearity, then that housekeeping protein is good for you, all right? Then stick to it. So for those researchers who have faced all these difficulties or inconsistent housekeeping protein result that make their quantitation later on, which a bit not, I'll say it varies a lot and they start to doubt what is happening in between. Therefore, we will always recommend them to go for total protein stains, because as compared to housekeeping protein where you just rely on one band, the total protein stain is, there's a lot of bands, means all the protein bands in the entire lane will be stained. Okay, based on the staining that you chose, it may be staining different regions, like for example, lysine, arginine, and so on and so forth. But they basically stain the total protein. So that's why it's total protein stains. Because you have more protein bands to become your denominator and to normalize your protein interest, it lessens the error and variability. Because whereas compared to one, you have one band over here, we have more, definitely more than one bands in total protein stain. So it lessens the error over here. And then here we're talking about variabilities because the reason being is the one versus more versus one. Because if you, let's say, have any certain treatment that changes the housekeeping protein expression, it wouldn't happen the same on total protein stain, because there's really, really small chance that the whole protein, the total protein expression will change in different condition. Sometimes at the normal situation, they will affect a set of protein, for example, but they will not affect the entire protein pool in that particular sample. That's why we say that total protein stains, it minimizes the error, biological and technical errors and variabilities. With that, because you're staining the total protein across the entire membrane, you can actually check or QC whether your loading is good or not. Let's say when you load this equal amount of protein across the entire membrane, you should see similar, I'll say, expression of the total protein. All right. So let's say if some accidentally spill some sample, then you will see that total protein volume or signal will be lessened a lot. Will be very obvious. Transfer efficiency also, you can QC it. Okay. You can also monitor the protein transfer, because let's say the transfer, it doesn't work well at the edge of the sandwich, you will see the total protein stain lesser in that particular area, or even bubbles or blocks of empty space. You will know it exactly when you see the image, right? But one thing for total protein stain is it normally uses fluorescent signals, okay? Fluorescent signals, because we want it to be reliable, right? And so it requires certain channel or digital imager to capture the signal, or certain excitation light sources. Right. So in this sense, with all these benefits, you can see the scientific community is starting to favor total.
Yep. Sorry to interrupt. How many slides do you have left?
I think, okay, I will speed up. So 5 minutes more, right?
Yes, sure. 5 minutes.
Yep.
Okay.
Let me grind through this. All right. Thank
Okay.
you. Thank you, Tani. Sorry, I'll take too much time. Indulge in the theory part. Right. So with this, total protein is now the new normal, right? So this is what you can see. Housekeeping protein, you see one band, okay? The one single band has served as housekeeping protein in this is fluorescent blot, but it works the same like in your chemi, but you won't see multiplexing here. Total protein normalization in this sense will be staining the entire protein over here. Okay, so that's why it provides better quantitation power and also better normalization. Right, so let's recap what we talk about. We'll talk about the principles of chemiluminescent Western, each step, every single step of them. Antibody detection, chemiluminescence, and fluorescent methods. Imaging part, we'll talk about the limitation of dynamic range X-ray film and digital imaging system. And digital imaging systems, CCD chip also, they have their own features as well. Normalization, we need to be careful. Okay, if you see something has been not constant in your housekeeping protein, do consider to change the total protein stains. It will help you a lot in the longer run. Right. So we are Azure Biosystems. We do have a lot of array of Western blotting reagents and equipment that fulfills your needs. All right. For example, we have gel tank system, reagent to stain total protein, or even chemi, Western, everything we covered. And imaging system over here, the Sapphire biomolecular imager and Azure imaging system. Okay, so let me just give a little bit of sales pitch over here. We have Azure imaging system, which is, we have a lot of models of them covering from basic, all can do gel documentation. So cover from gel documentation up to chemiluminescent signal detection, and also fluorescent, RGB, near infrared, or combo of both them, which is 600, our highest range. Right, so there's just a specification of these models, like the camera and also the cooling system, the filter wheels, and so on and so forth. Right, right. Azure Imaging System 300 is the one that we will go for if you want to do chemiluminescent Western imaging, because it has better camera, okay, 9 megapixel, and you can do merging with color marker, so it's easier for you to identify the protein bands to know that that is a specific bands, right? So you can do quantitative chemiluminescent imaging as well. This is what we try to give you, because you can detect the saturation, which is, I think, commonly you will see that there's one of the advantages for using digital imaging system to detect capture chemiluminescent Western. All right. Right, so here's an example of publication that we have been published using Azure Imaging System 300. We have the one, these are just, I got it this year and last year. Some representative publication. We have one from Cell Report, which is like impact factor 9.4. We have the PMC Alzheimer research with impact factor 6, and we have the Frontiers and Molecular Neuroscience, like impact factor 3 point something, and the MDPI, if you're familiar with this publisher. Okay, right. So you can see that a good, I'll say, Western blot imaging system do actually enhance and support your scientific findings. And let me introduce you to the next one, will be Azure Imaging System 600, which is a higher range of Azure Imaging System. It can do both near infrared and RGB fluorescence, and also chemi and all the other gel documentation and protein gel as well. Here RGB, we use LED because RGB itself, they have very distinct separated spectra, so it doesn't need to go for laser to properly separate the wavelength. The near infrared range, we will have laser to excite them, specifically excite them. So that's why the pairing will be good. So we do this with this, we can do up to 5-plex. Okay, but we at the moment, because of the combination of primary and secondary antibody, we at most can do 4-plex only. All right, yeah. So this is the very common DNA gel band. Okay, DNA gel image. Western blot gel, Western blot image with color marker, frozen Western blot over here. This is basically multiplexing, and protein currency, and then bioluminescent Patrick. I'll say this is what microbe. Okay, and cell zebrafish over here. Anything comes with frozen because it's a full spec one, frozen, and also autofluorescence from the animal or even the plant itself, you can capture with the system, right? So there's a lot of things in your research that you can utilize this, and this is just a very snippet about how they use this system for. Some researchers, they even use it to capture plant, which we never think about it of, right? Right, so here's some representative publication. We have the one from Advanced Biosystem, which is like Q1 in bioengineer, right? This is capturing commensal protein gel imaging, and we have a publication in, I think this year or last year, from in Journal of Immunotherapy of Cancer, which impact factor 13.75. So this is pretty impressive. Right, so of course, this imaging system comes with analysis software where you can do for 2D densitometry, automatic Leng detection and band, molecular weight analysis, quantity calibration, multiplex analysis, dendrogram, colony counting, arrays and plates, and so on and so forth. Right, so Azure Biosystems, we have like I say, we have imaging solution for every labs, and especially protein application, down to very Western blot reagents and or microplate reader, basic gel imaging, all these models, up to chemi, fluorescent, and full spec fluorescent, NR, RGB fluorescent, in the higher spec, which is our Sapphire biomolecular imager, that we really proud of, and will still have a lot of new application coming up using this. But the researchers are doing their creativity there. Yeah, so all my information or the presentation information will be available in this Western Blotting guidebook. Okay, and please, if you can do it, download it from this link. Okay, www.azurebiosystem.com/guidebooks. Everything will covered in this guidebook, talking about the principles and what's the idealism behind it, why you want to do this step, and then why you want to put methanol, or let's say why you want to watch out. They've been explaining every single thing, covering both chemi and fluorescent Western, and all the application notes available. Right, so I am Kai Leng. Thank you for your time today. So if any inquiries, do contact us via the emails listed above, and also Dani, which is our MC today, who is our MC today. Yeah, thank you everyone. So I'll pass the floor back to Dani.
Okay, thank you for your great presentation, Dr. Kai Leng Tan.
No problem.
And yeah, let me share my screen. Yeah, so there is so many information that we can get from Dr. Kai Leng Tan presentation, especially everything about Western blot and the support that we can provide in development in Western blotting. So we already know that Azure Biosystems products are really suitable for protein detection and analysis using Western blotting. And yeah, that was very good presentation. And thank you, Dr. Tan.
No problem.
Yes, and so interestingly, we see that there are many already several questions waiting for you, but we need to wait first because it will be answered during the Q&A session, Dr. Kai Leng Tan. Yeah,
Sure.
thank you. So, Bapak Ibu sekalian, kembali kami ingatkan untuk pertanyaannya itu bisa diajukan dengan menggunakan tombol Q&A dan juga fitur chat yang tersedia dalam platform Zoom yang disediakan. Kemudian saat mengajukan pertanyaan, mohon disebutkan ditujukan ke pembicara yang mana, dan disebutkan juga nama beserta instansi Bapak Ibu sekalian. Baik, Bapak Ibu sekalian, kita melangkah ke materi selanjutnya mengenai application of Western blot and biomedical research yang akan disampaikan oleh Dr. Febriana dari Universitas Indonesia. Dr. Febriana merupakan dosen dari Departemen Biokimia dan Biologi Molekuler dari Fakultas Kedokteran Universitas Indonesia. Beliau merupakan lulusan strata 1 dan strata 2 dari Universitas Indonesia, kemudian dilanjutkan dengan program doktoral di tahun 2018. Beliau saat ini merupakan dosen Departemen Biokimia dan Biologi Molekuler di Universitas Indonesia, kemudian dosen tetap program Magister Ilmu Biomedik, dan juga Ketua Cabang Jakarta Perhimpunan Biokimia dan Biologi Molekuler Indonesia. Dan tentunya terdapat beberapa internasional publikasi yang sudah dipublikasikan oleh Dr. Febriana. Baik, selamat siang menjelang sore, Dr. Febriana. Bagaimana kabarnya, Dok?
Baik, Pak Yahya.
Baik. Untuk presentasinya, Dr. Febriana, apakah masih butuh persiapan atau sudah bisa kita mulai saat ini, Dok?
Sudah bisa, saya rasanya. Suara saya cukup jelas tidak ya?
Sudah cukup jelas, Dok. Ya. Baik, Dr. Febriana, jika sudah bisa dimulai kepada Dr. Febri, kami persilahkan, Dok.
Ya. Ya, terima kasih Pak Dani. Assalamualaikum warahmatullahi wabarakatuh. Sebelumnya saya mengucapkan terima kasih ya kepada Bu Lianita, Marketing and Technical Support Manager dari PT Biosm atas kesempatan pada
Sama-sama.
siang hari ini untuk sharing tentang aplikasi dari Western Blot. Also, thank you to Dr. Leng for very interesting presentation. Baik, pada kesempatan ini saya akan sharing sedikit beberapa riset-riset yang berjalan di departemen kami, Departemen Biokimia dan Biologi Molekuler di FKUI, terkait dengan penggunaan Western Blot. Berikut adalah outline dari materi yang akan saya sampaikan. Tadi sudah dijelaskan dengan sangat clear oleh Dr. Leng ya, terkait dengan prinsipal dan teknik dari Western Blot serta berbagai aspek yang berperan dalam menghasilkan Western Blot yang sangat baik. Jadi memang Western Blot ini peranannya sangat penting gitu ya, terutama pada riset-riset biomedik maupun pada riset klinik, terutama untuk identifikasi protein yang spesifik, baik berdasarkan ukuran maupun pengikatan terhadap antibody. Tiga elemen utama yang paling penting adalah proses separasi dari protein berdasarkan ukuran, kemudian transfer ke solid support, tadi sudah dijelaskan oleh Dr. Leng ya, bisa PVDF maupun nitroselulose dan plus minus dari masing-masing. Kemudian identifikasi dari protein melalui konjugasi dengan antibodi primer maupun sekunder untuk proses visualisasi. Ini adalah beberapa hal yang saya highlight terkait dengan separasi, beberapa hal yang cukup kritikal ya, mulai dari proses ekstraksi, fraksinasi maupun purifikasinya, kemudian juga sampel, preparation-nya, loading, kemudian konsentrasi sampel itu juga sangat berperan. Kemudian prosedur transfer tadi juga sudah dijelaskan dengan sangat clear ya. Kemudian juga metode deteksi, berbagai pilihan untuk deteksi, tapi yang utama menurut saya validasi dari antibodi primer, baik spesifisitas maupun selektivitas dari antibodi yang kita gunakan. Baik, saya lanjut dengan aplikasi dari Western Blot. Ini adalah sharing dari beberapa penelitian di tempat kami. Yang pertama ini adalah protein identifikasi atau identifikasi protein dan juga evaluasi dari reaktivitas protein yang dihasilkan tersebut terhadap beberapa kelompok dari serum pada manusia. Pada penelitian ini yang kami fokuskan adalah protein P24 dari subtipe B virus HIV-1. Dari gambaran skematik dari virus HIV ini kita bisa lihat bahwa protein P24 merupakan protein struktural, sedangkan protein-protein cukup banyak pada virus HIV, antara lain protein envelope, ada GP41, GP120, kemudian juga protein matriks, P17, dan seterusnya. Nah, pada penelitian ini dilakukan konstruksi, transformasi, kemudian juga ekspresi dari P24 pada E. coli BL21. Kemudian hasil ekspresi ini dilakukan purifikasi dengan kromatografi afinitas, dan kemudian dilakukan uji reaktivitas dengan menggunakan Western Blot. Nah, ini adalah gambaran uji konfirmasi dari hasil ekspresi protein P24. Pada bagian ini adalah konfirmasi dengan menggunakan SDS patch. Tampak pada gambaran ini adalah lisatnya, ya, lisat sebelum dilakukan purifikasi. Kita lihat masih cukup banyak campuran protein yang terdapat pada lisat. Kemudian pada len yang berikutnya ini adalah hasil purifikasinya. Kita lihat tampak protein tunggal ya. Kemudian dari hasil purifikasi ini kita lakukan identifikasi dengan menggunakan uji Western Blot. Uji Western Blot ini digunakan yang kolorimetri ya. Kita lihat baik pada lisat maupun pada hasil purifikasi muncul pita yang tunggal ya pada posisi sekitar 25 kilodalton. Nah, kemudian setelah diperoleh protein yang hasil purifikasi tadi, kemudian dilakukan uji reaktivitas. Uji reaktivitas ini dilakukan pada beberapa serum. Ya, yang pertama, pada gambar yang pertama ini adalah diuji dengan serum yang sudah terkonfirmasi dengan HIV positif. Kita lihat di sini dari 7 serum yang diujikan, semua menghasilkan pita tunggal, ya, pita protein tunggal, walaupun dengan ketebalan yang berbeda-beda. Mungkin ini terkait dengan konsentrasi antibody dari masing-masing serum tersebut. Kemudian kalau kita lihat pada kontrol positif, tampak pita yang cukup clear, kemudian juga kontrol negatifnya tidak tampak adanya pita. Sedangkan pada kelompok yang kedua, ini adalah pada gambar B ini, protein P24 kita reaksikan dengan serum pada orang sehat. Dan bisa kita lihat di sini bahwa tidak muncul pita pada posisi sekitar 25 ya, jadi memang tidak reaktif ya, tidak ada cross reaksi gitu ya. Nah kemudian yang ketiga ini dilakukan uji reaktivitas terhadap serum yang indeterminate ya. Pada HIV kita mengenal istilah serum indeterminate, yaitu serum yang belum konklusif ya, jadi masih harus dikonfirmasi lagi dengan uji konfirmasi. Pada serum indeterminate ini tampak beberapa sampel individu itu memberikan reaksi ya, walaupun lemah gitu ya. Sedangkan pada kontrol positifnya tampak pita yang tebal, sedangkan kontrol negatifnya bersih, tidak tampak adanya pita. Berikutnya adalah perbandingan ekspresi protein dari berbagai organ. Pada penelitian ini fokusnya adalah pada ekspresi myoglobin. Ya. Ini diambil pada green turtle atau tukik. Nah, myoglobin ini adalah salah satu dari member globin family. Ya. Kalau kita lihat banyak globin family antara lain hemoglobin, sitoglobin, neuroglobin, dan seterusnya. Myoglobin ini termasuk protein globular yang dia mengandung gugus Hem ya. Nah, paling banyak dia diekspresikan terutama pada otot polos dan peranannya terutama dalam oxygen storage. Nah, pada penelitian ini ditujukan untuk menentukan apakah myoglobin ini terekspresi juga pada jaringan-jaringan yang non muscle ya. Sehingga diuji pada beberapa organ gitu ya. Pada lane yang pertama ini adalah otak, kemudian yang kedua jantung, yang ketiga adalah hati. Nah, ternyata pada saat dibandingkan di antara ketiga organ ini yang terekspresi myoglobin ini pada hati, sedangkan pada organ yang lain tidak tampak terekspresi. Ini adalah kontrol positif ya, yang menggunakan jaringan otot dari tikus ya. Berikutnya adalah perbandingan antara ekspresi protein dengan ekspresi dari mRNA. Fokus pada penelitian ini adalah pada hipoksia inducible factor 1 alpha atau HIF1 alpha yang dia terutama berperan pada kondisi hipoksia. Kebetulan di departemen kami, hipoksia dan oksidatif stres merupakan salah satu fokus penelitian yang cukup cukup banyak diperdalam gitu ya. Dan pada penelitian ini adalah menguji pola ekspresi dari HIF1 alpha yang pada model hewan coba, ya, hewan coba tikus yang dilakukan dibuat model menjadi hipoksia kronik yang sistemik dengan menggunakan suatu hipoksia chamber. Nah, kemudian dari ginjal tikus tersebut kemudian dilakukan isolasi dan mRNA-nya diukur menggunakan real-time PCR, sedangkan proteinnya diukur dengan menggunakan Western blot. Nah, dari hasil Western blot tampak di sini pita-pita yang muncul, gitu ya. Kemudian dilakukan pengukuran secara semi kuantitatif. Kita lihat di sini dari beberapa kelompok, ada kelompok kontrol, kemudian hipoksia hari pertama, ketiga, 7, kemudian 14, dan tampak di sini pita yang paling tebal ada pada kelompok hipoksia yang 7 hari, dan kemudian menurun pada kelompok berikutnya. Kemudian dari hasil tersebut dilakukan uji korelasi. Uji korelasi ini dikorelasikan dengan PO2 atau tekanan O2 pada plasma terhadap intensitas dari protein pada uji Western blot, dan ternyata terdapat korelasi yang negatif kuat ya antara PO2 dengan HIF1 alpha. Sedangkan pada korelasi antara mRNA, ekspresi dari mRNA HIF1 alpha terhadap protein tampak terjadi kesesuaian ya, korelasi yang positif kuat. Ya, berikutnya adalah ekspresi protein pada proses regenerasi. Model yang digunakan pada studi ini adalah model cicak ya, untuk dilihat proses regenerasinya. Fokus dari penelitian ini terutama adalah pada HIF1 alpha dan HIF2 alpha. Sebenarnya dilakukan pengamatan beberapa metode gitu ya, mulai dari visual, pengamatan secara panjangnya gitu ya, diukur panjangnya, kemudian juga diamati secara visual mulai dari hari pertama sampai hari ke-30. Kemudian juga dilakukan observasi secara histologi menggunakan sediaan HE, kemudian juga ada imunohistokimia. Ya, berikutnya dari beberapa kelompok yang diamati tadi, mulai dari hari ke-1 sampai hari ke-30, dilakukan pengukuran ekspresi dari HIF1 maupun HIF2 alpha, baik pada level mRNA-nya dan juga pada level proteinnya. Kita lihat di sini, ini adalah ekspresi dari mRNA HIF1 alpha, sedangkan ini adalah ekspresi dari mRNA HIF2 alpha. Sedangkan pada gambar yang ini adalah beta-aktin sebagai housekeeping protein, kemudian HIF1 alpha, dan ini dibandingkan pada beberapa kelompok pengamatan. Gitu ya. Kemudian dari hasil uji Western blot ini dilakukan pengukuran data secara semi kuantitatif, diukur intensitas dari band atau pita protein tersebut, kemudian dilakukan perhitungan rasio terhadap housekeeping gene-nya, sehingga didapatkan rasio protein yang terekspresi baik HIF1 alpha maupun HIF2 alpha. Nah, pada gambar yang sebelah tampak bahwa puncak HIF1 alpha itu pada pengamatan hari ketiga, sedangkan pada HIF2 alpha puncak protein terdapat pada hari ke-8. Ya, berikutnya adalah identifikasi dan juga konfirmasi dari enzim yang diisolasi. Enzim yang diisolasi adalah enzim reduktase, diambil dari source-nya adalah hati dari sapi. Setelah dilakukan isolasi, kemudian dilakukan purifikasi. Purifikasi ini dilakukan menggunakan kromatografi afinitas. Kita lihat di sini dari hasil kromatografi afinitas terdapat 2 peak ya, peak yang A dan juga peak yang B. Nah, dari kedua peak ini kemudian dilakukan analisis lebih lanjut ya. Kemudian dilakukan SDS page, ini semi denatur, kemudian diwarnai dengan pewarnaan silver staining, dan tampak di sini pada yang lisak kelompok yang peak A, peak B, dan juga kontrol yaitu diaforese. Kemudian dari hasil SDS patch dilakukan uji Western blot. Kita lihat di sini antara T-sat kelompok yang peak A dan peak B tampaknya yang memang terdapat protein yang atau enzim yang diisolasi adalah pada yang peak A ya, sedangkan peak B tidak tampak adanya pita protein. Sedangkan pada lisat masih tampak cukup tebal. Kemudian dari hasil isolasi tadi, setelah dikonfirmasi, kemudian dilakukan uji aktivitas dari enzim tersebut, yaitu enzim reduktase. Ya. Terakhir berikutnya adalah konfirmasi dari konsentrasi protein secara kuantitatif. Studi ini sebenarnya studi yang cukup panjang, menggabungkan antara in silico dan studi in vitro. Tujuannya untuk mendapatkan active compound yang sifatnya natural, gitu ya. Jadi kalau kita lihat di sini dimulai dari ada 7 compound yang diuji, kemudian diseleksi, dilakukan prediksi model, kemudian dari hasil seleksi tersebut kemudian didapatkan satu bahan aktif yang dianggap paling potensial, yaitu andrografolid. Nah, pada andrografolid ini juga dilakukan molecular docking, ini molecular dockingnya, dan kemudian setelah didapatkan kandidat yang paling potensial, kemudian baru dilakukan uji in vitro. Nah, uji in vitro dilakukan pada stem cell kanker payudara. Fokusnya terutama adalah pada survivin, karena pada stem cell kanker payudara, ekspresi survivin ini sangat tinggi, dan kita tahu survivin ini merupakan anti apoptosis, ya, sehingga kemudian dijadikan target pada penekanan survivin dengan menggunakan andrografolid tadi. Dan kita lihat di sini ada beberapa konsentrasi dari andrografolid yang dicobakan. Kemudian selain survivin juga diuji pada caspase 3 maupun caspase 9. Ini adalah hasil dari ekspresi mRNA. Kemudian ini adalah hasil ELISA dari survivin, baik survivin total maupun survivin yang terfosforilasi. Nah, kemudian dari sini dilakukan uji konfirmasi dengan menggunakan Western blot, baik untuk survivin, phosphorylated survivin, caspase 3, caspase 9, dan menggunakan housekeeping protein beta-aktin. Ya, tampak di sini ini adalah kontrol, kemudian ini adalah perlakuan konsentrasi dari andrografolid yang berbeda-beda, gitu ya. Tampak juga pada berbagai konsentrasi yang berbeda, ternyata juga menghasilkan pita yang ketebalannya juga berbeda. Ya, demikian yang bisa saya sharing dari penelitian-penelitian yang terdapat di departemen kami. Terima kasih. Saya kembalikan kepada Pak Yahya.
Baik, terima kasih atas presentasinya Dr. Febriana. Sangat menarik materi yang disampaikan, sehingga bisa didapatkan ilmu-ilmu berharga yang bisa digunakan dalam melakukan Western blotting, khususnya dalam dunia biomedik. Namun kita tahu bahwa penerapan Western blot yang kita ketahui tidak hanya sebatas di situ saja, namun begitu banyak penerapan-penerapan yang di luar sana yang bisa diaplikasikan dari Western blot ini. Jadi Bapak Ibu sekalian, sudah disampaikan materi dari Dr. Febriana. Jika Bapak Ibu sekalian memiliki pertanyaan baik terhadap Dr. Leng ataupun Dr. Febriana, sekali lagi bisa disampaikan melalui Q&A button dan juga chatnya. Nanti pertanyaannya disebutkan akan ditujukan ke pembicara yang mana, dan disebutkan juga nama beserta instansi dari Bapak Ibu sekalian. Sembari mengumpulkan pertanyaan dari para peserta webinar hari ini, sedikit akan kami tunjukkan video dari Azure Biosystems, merupakan video demo ataupun informasi mengenai produk dari Azure Biosystems. Ya.
Now, there is one system for all your Western blot imaging needs, the Azure Biosystems C-Series. Fluorescent Westerns and chemiluminescent Westerns are captured as crystal clear images, quickly, with high sensitivity. C-Series systems are easy to use, offer multiple detection modes, and a complete upgrade path, all in a small footprint. The Azure C600 is the only imaging system that offers 3 modes for Western blot imaging in a single system: laser infrared fluorescence, LED visible fluorescence, and chemiluminescence. Take advantage of laser infrared fluorescent imaging for quantitative Western blot analysis with high signal-to-noise. Detect low abundance proteins with sensitive chemiluminescence, or 3 proteins on a single blot with RGB visible fluorescence detection. Or capture images of your DNA and protein gels.
Azure Biosystems was founded because we believe in creating products that help scientists improve how they design their experiments and make it easy for them to obtain their results. Our team of scientists and engineers have been designing, manufacturing, and supporting Western blot imaging applications for over 20 years. We've observed the emerging trends in the marketplace and have designed a series of instruments that deliver real benefits to the bench scientist.
Fluorescent Westerns will continue to grow in use as researchers take advantage of their quantitative accuracy. But we also recognize that many users aren't ready to give up chemiluminescence, nor do we think that they should have to. There are many times where the assay dictates that chemiluminescence is your best detection chemistry. Researchers today need a system that can image both. Azure Biosystems is the only manufacturer to offer laser-based near-infrared fluorescence, LED-based visible fluorescence, and chemiluminescent detection all in the same system. Capturing images in any of our application modes is easy. Place your sample in the system, select your sample type, press capture. The system is controlled through a simple touchscreen tablet interface. Researchers have really embraced our C-Series line of imaging systems because they produce high-quality images and they're extremely easy to operate. It just works.
The Azure product line culminates in the C600 system, the only imager on the market that combines an 8.3 megapixel camera for crisp, high-resolution images and fast imaging, LED excitation of dyes in the visible range for 3-color multiplexed Western blots, laser diode excitation of near-infrared dyes for multiplexed Western blots with improved signal-to-noise, a compact design with an integrated touchscreen tablet that delivers an unmatched customer experience and ease of use. Dual focus technology. Image samples in perfect focus and with optimal lens settings without having to touch the camera. And standalone analysis software for true quantitative applications. The C-Series is fully upgradable from the C200 for imaging DNA and protein gels all the way to the C600, offering chemiluminescent detection, RGB fluorescence, and infrared fluorescence, all in the same system. Choose the model that is right for you and never worry about needing another system as your methods evolve. Leave the darkroom behind. You can easily image your gels and blots by any method you choose with the Azure Biosystems C-Series. To learn more, contact Azure Biosystems at www.azurebiosystems.com.
Danny, I think you're muted.
Ya, baik Bapak Ibu sekalian, itu tadi video dari Azure Biosystems dan melangkah ke agenda berikutnya yakni sesi tanya jawab. Di sini saya melihat sudah ada beberapa pertanyaan, sekitar 8 sampai 9 pertanyaan yang sudah diajukan. Tapi saya akan ke Dr. Leng terlebih dahulu untuk menjawab pertanyaan dari para peserta. So, Leng,
Yes.
yeah, I think we have around 5 or 6 question for you. On the Q&A box, do you want me to read it, or do you want to directly answer the question?
I think it's fine. I can directly answer the question.
Yeah, and from the chat box, I will read for you.
Okay,
question.
sure.
Yeah.
All right, sure.
Okay.
So I do have them sorted in the Word document at the moment.
Yes.
Do you want me to answer in the Q&A panel first, or how should we go? Okay.
Yeah, I guess
mean-
can I just read out the question and then give the answer?
Okay.
Yeah.
I'll go with the one that I captured from the chat room and also the Q&A panel first. All right. So the first question from the Q&A is from the Q&A panel, which is, when is qualitative Western blot needed and when quantitative needed? So the qualitative Western blot is needed, which is you just want to see whether the protein of interest is there or not. All right, so that is qualitative. If you just want to check there, you can go for either colorimetric, like the one that Dr. Febriana showed, which is super good, is everything optimized, it will be very cost effective, okay? Or you can go for chemi. That means you just need to see, that the protein A is there. Yes, that's it. But quantitative, where you have multiple samples and multiple groups or multiple factors. For example, you want to compare treated and non-treated, and also and also different tissue or different stages, then you might need to go for quantitative, because you need to accurately compare. For example, the treated group has, let's say, 2-fold expression higher than the untreated control. You want the exact protein expression data. So that's why we say go for quantitative if you need to provide that kind of information. But if you just want to check, this protein has let's say a novel protein. This sample has a novel protein, then you can go for qualitative, just checking yes or no, right? Okay, I hope I answered your question. Because this is anonymous submission, if you need me to further explain, please further extending question in the Q&A panel. All right? Okay, so the next one.
Bapak Ibu sekalian, kalau misalkan masih ada pertanyaan lanjutan bisa nanti diajukan lagi. Baik, untuk melangkah ke pertanyaan berikutnya. So next question from Nikan Rahmagani. So hi, I would like to ask regarding which part is the most important phase in Western blot as an analytical approach for detection protein, which requires greater attention so that we can avoid protein detection failure while utilizing Western blot.
All right, so I think I can give a little bit of experience over here. Western blot itself, any experiments that you do for your scientific research, everything are important. Down to the control groups that you set up, experiment, how you do your experiment before you prepare the sample itself. That's like Dr. Febriana's slide say, Your sample preparation also very important. Then when you do your Western blot, the way, of course, the pipetting skill must be good, okay? And then you use the correct lysis buffer, load it properly, okay? Gel electrophoresis run properly across the entire gel, okay? Nothing crooked. Then you transfer it properly. So every single step actually have their own QC, okay? So it's science is like this. It's not easy, okay? So that's why I do think Nicole to explain that actually every single step is very important. Nothing to say that you know, or most important phase, no. Actually, most of them, all of them are important, because any single step that you, let's say you misuse or something wrong in between, or you mix up or something, the whole experiment or the result you get by the end of the day will not, you cannot use. Yeah, so that's why a lot of researchers or a lot of students, they will be repeating their experiment again and again. And my personal advice is, do pay attention to details. Every single thing you have your lab notebook, okay? Note down, let's say today your blocking buffer is a reused blocking buffer. It's not a fresh one. Do jot it down. Because let's say this experiment you run has a little bit of problem, then you know that the issue might arise from the reused blocking buffer. Okay, so this is just some tips and tricks. Okay, the general answer is pay attention to all of them. Every single thing is important, right? Okay.
Okay. So next question from Apriliani Irawan. Hi, I want to ask about primary antibodies. In my lab, I have a custom protein I want to detect using Western blotting. How do I obtain a customized primary antibody that can detect my protein?
Yep. All right. So this one, in my own experience, I use commercial antibodies. Okay. But let's say if you have a protein that no one has, no antibody commercially available, the only way you want to detect it is to produce your own antibody. That's the only way to do. If you do check certain publication, some of them, they produce their own antibody, some of the labs. It's a very lengthy and I'll say very it's a full process, because manufacturing antibody is not easy. You need to select the clone to produce antibody for you. Yeah, so I could not really go into details on how to do that, but I do say that customized primary antibody, I believe what you say is not commercially available. If it's not, then you need to produce your own primary antibody. Yeah.
Yeah. And next question from Vina Nurverdianti. Thank you very much, Dr. Kaileng Tan, for your amazing presentation. If I would like to validate some proteins like 10 kilodalton to 30 kilodalton by Western blot, what should be the ideal transfer condition, volt, milliampere, or time period for detection of the protein?
All right. So I believe that since you are doing very, very low molecular Western blot, okay, so the first thing you need to choose, I believe that the gel that you use, that's supposed to be at least 12% and above, okay, because you need to separate them properly. All right. Wet transfer is the only way to go. Don't do semi-dry, wet transfer, okay? Wet transfer wise, I'll say the voltage and milliampere don't more, okay, I'll say like this, 70 voltage, less than 2 hours. We can try it 70, 20, a 70 volt, 2 hours, try it. If it's try it once, okay? If compared to your wherever you setting that you have used, if you see the bands are all okay, then stick to it. All right. So if it's not, then I'll say tweak the voltage or tweak the time. Do not more than 2 hours. Do not set more than 2 hours. Or another way is you go for 15 volts overnight. Either that, one or another. Okay. 70 voltage, don't more than 2 hours. 15 volts overnight. So overnight is something like this. You might need to determine a time frame for that. For example, 8 hours, 16 hours, like that. So this is up to you. I personally do not really have not done low molecular weight before, but based on my friend's experience, what they do is they do overnight at low 15 volts, right? So if you need information, I may actually dig out their paper and send it to you, and you can refer to it. Okay.
Okay. Yeah, we still have 4 questions for you, Dr. Kailan. The first one
Okay.
come from Nur Hasana from University Segmentation. The question is, can we test the protein with Western blot in the form of crude extract protein, or does it have to be pure?
Okay, crude. I would like to know what is the crude protein means, because all the protein lysate they will get from either cell or tissues are mostly crude. Okay, by the means of that, you will still need to either do denaturing step, heat it up with a loading buffer. The loading buffer itself contains glycerol and colors and so on and so forth. That will actually let the sample sink inside the well when you load it in the gel. Right, so those are actually quite important. And sometimes because of the lysis buffer you use, you might need to dilute it so the remaining of the detergent or so on and so forth will not interfere the running and transfer later on. Yeah, I'll say you, sorry. Okay, can we test the protein with Western blot in the form of crude? Yes, you can. You can test it in the form of crude extract protein. Okay, purified protein depends or how to say you want to purify it. You want to purify it free of the lysis buffer, or you want to purify it free of certain reagent. Okay, that is depending on your experiment. If your experiment, let's say you're studying certain lipid or studying certain modification, then you might need to go for certain let's say crude extract or certain post-processing to make it, I wouldn't say pure, but I'll say post-processing step like that before you run it to Western blotting protocol.
Okay, thank you. Next question from Surya. In case detection of the outer membrane protein of bacteria, can we detect the protein from whole cell bacteria, or we have to extract the protein first?
Okay, so you have a group of colony, bacterial colony, right? So when you do like extraction, you extract the entire protein. So it's either from the membrane or from the cytoplasm and the nucleus. Everything will be inside. Unless specific region do you want to do, let's say cytoplasmic protein, then you need to do fractionation Western blot. Okay, but in this sense, you want to detect, in your case, you want to detect outer membrane protein and bacteria, you can just extract it from the entire bacteria. You don't need to do fractionation. Fractionation is for comparing the membrane versus cytoplasm versus this. Okay, you just need the outer membrane. So we calculate that everything as a whole, since you just extract the whole bacteria colony. That's it. Yeah, but do remember my suggestion here is based on limited context that you provide here. It may not be true. So the most important thing is refer to your PI or discuss with someone who at the similar field or niche with you. Okay, refer to publication and literature. That is something that is will guarantee you that you're going to have a successful experiment later on. Right.
Okay. Next question from Andriana Pope Estania. The question is, if we are using fluorescence detection method, is there any way to reprobing the membrane? Do you think the stripping method that commonly used in chemiluminescence imaging will work when we are working with fluorescence?
Okay, so in fluorescence detection method, the reason people go for fluorescence is they just don't want to strip and reprobe. That is the ultimate thing. They want to do multiplexing. They do not want to strip and reprobe. All right. So, but if you, let's say you want to strip and reprobe it again, I personally never do it, but I feel that there might have people doing it. But at the same time, I do feel that strip and reprobe itself do, how to say, counteract the benefits of the fluorescence detection method. Because the reason you want to do fluorescence is you do not want to strip and reprobe, right? So yeah, it's contradicting to each other. Yeah. The common stripping in chemiluminescence, would it work in fluorescence? I never tried it, so I cannot really vouch for about it. But I do say that for those who done fluorescence Western blot so far that I know of, they don't strip and reprobe, right? The reason being is, first thing, you want to either you squeeze all the combo, the detection proteins of interest into a single blot, let's say up to 4-plex, okay? Okay, up to 4-plex you want to do 3+1, either 3 plus housekeeping or 3 plus total protein, right? So 3 proteins at the same time at the same blot. Yeah, that's what you want to do. If you, let's say you have 10 protein of interest, okay, then you have to put it into 3 separate blots. Yeah, we do not encourage strip and reprobe on the membrane, because even though no matter how hard you take care of the situation, you know, the buffer and the condition and washing, there will still be risk that you lose protein in between. So since you're going for fluorescent detection method, might as well just stick to the stringent part of it, stick to it and do it just like that, 1 multiplex at a time. Then you know that in this setting, this blot, everything is nice. So you won't be, you know, doubting your result later on. Okay? Yeah,
yeah. Another question from same person, from Andrian. The fluorescence detection method allow us to detect multiple proteins at the same time. Does it mean we have to mix all the primary antibodies that we want to incubate for overnight incubation?
Yes. So multiplexing means you can incubate, like normally you do one at a time, will be 1 primary antibody overnight. So multiplex means you mix out all the primary antibody, incubate it overnight. Yeah. Yes, that's how you do it.
Okay, thank you Dr. Leng for your answer. Bapak Ibu sekalian, jika masih ada pertanyaan, nanti bisa diajukan lagi. Saat ini kita akan melanjutkan ke Dr. Febriana terlebih dahulu. So thank you Dr. Leng. I will go to next question for Dr. Febriana. Kami ingatkan lagi untuk mengisi form feedback kepada Bapak Ibu sekalian untuk nantinya sebagai daftar hadir dan juga bahan untuk pembuatan e-sertifikat yang akan kami kirimkan ke Bapak Ibu sekalian. Baik, Dr. Febriana, ini ada 2 pertanyaan, Dok, dari peserta webinar. Yang pertama dari Priscilia Felicia, mahasiswa baru S2 Ilmu Biomedis UI. Saya ingin bertanya kepada Ibu Febriana, terutama pada berbagai penelitian Ibu menggunakan Western blotting untuk berbagai kebutuhan deteksi. Masing-masing tentunya mendeteksi protein yang berbeda-beda dengan epitop yang berbeda-beda pula. Saya ingin bertanya bagaimana cara Ibu mendapatkan antibody primernya? Kemudian apakah Ibu beli atau desain sendiri dulu atau bagaimana? Juga apakah Ibu lebih sering menggunakan antibodi monoklonal? Jika iya, kenapa ya Bu? Silahkan Dr. Febri.
Ya, terima kasih Pak Yahya. Terima kasih Priscilia. Ya baik, terkait dengan penggunaan antibodi, betul, target proteinnya berbeda-beda, kemudian juga epitopnya pasti berbeda. Biasanya saya menggunakan antibodi primer komersial, ya, beli, namun juga harus diingat, pada saat membeli antibodi komersial, kita harus memastikan bahwa protein target kita itu memang benar akan dikenali oleh antibodi tersebut. Jadi biasanya dari apa namanya spesifikasinya ada penjelasan di situ, dia bisa digunakan untuk apa dan dia mendeteksi pada epitop yang mana. Kemudian juga kadang-kadang kalau ada protein-protein yang misalnya yang terfosforilasi atau yang tidak dan seterusnya, nah itu kadang-kadang ada beberapa yang berbeda. Kemudian antara poliklonal dan monoklonal. Tadi Dr. Leng juga sudah menjelaskan penggunaan antibodi poliklonal maupun monoklonal. Memang masing-masing ada plus minusnya gitu ya. Kalau kita menggunakan antibodi monoklonal sangat spesifik, tapi risikonya kadang-kadang kalau apa namanya ternyata protein target kita tidak dikenal oleh antibodi tersebut, maka tidak akan terdeteksi gitu ya. Jadi biasanya saya lebih sering menggunakan poliklonal. Tapi kalau misalnya memang yang komersial ada monoklonalnya dan sesuai dengan protein target, itu sebenarnya tidak masalah. Kita juga bisa saja pakai antibodi monoklonal. Jadi biasanya sih per kasus ya kita lihatnya. Pada studi ini apa yang kita lakukan, kemudian target kita seperti apa, dan ketersediaan antibodi komersialnya seperti apa gitu. Saya rasa itu Pak Yahya.
Baik, terima kasih dok pertanyaannya. Masih ada 2 pertanyaan lagi dok. Good afternoon, Mrs. Dr. Febriana. My name is Zikri from PPI SHP. I have a question. How the method and result of reaction protein of human sera? If we try to fuse a sample, can method do it? Mungkin Dr. Febriana bisa menyampaikan jawabannya dalam bahasa Inggris ataupun dalam bahasa. Silahkan.
Ya, terima kasih Zikri. Untuk pada studi yang menggunakan human sera, yang kami lakukan adalah studi reaktivitas. Jadi memang yang kami uji adalah protein yang dihasilkan atau diekspresikan. Nah, pada saat membandingkan, kami menggunakan beberapa human sera. Ini ada kami setelah ditransfer, kemudian dilakukan pemotongan per line, gitu ya. Kemudian masing-masing direaksikan dengan human sera yang berbeda. Jadi ada semacam chamber di lab kami ya, yang kita bisa gunakan per line untuk uji immunoblotting, sehingga masing-masing bisa kita isi dengan human sera yang berbeda. Kalau terkait dengan pada fish, saya tidak terlalu jelas ya, maksudnya apa yang mau dideteksi. Kalau pada studi yang menggunakan human sera ini berperan sebagai antibody gitu ya, primary antibody gitu ya, sehingga tergantung pada desain penelitian yang pada fishes tadi gitu ya, apakah yang mau dideteksi adalah antibody yang dihasilkan dari fish tersebut. Kalau memang iya, saya rasa bisa gitu ya. Hanya nanti tinggal protein targetnya apa gitu. Saya rasa itu Pak Yahya. Terima kasih.
Baik dok. Kemudian pertanyaan selanjutnya datang dari Vina Nur Ferdianti. Terima kasih banyak Dr. Febriana untuk presentasi yang sangat baik. Apakah memungkinkan dok apabila kita mengaplikasikan metode ini dalam penelitian yang sumbernya itu berasal dari urin? Apakah ada pengalaman atau informasi research terkait hal ini? Terima kasih banyak dokter. Silakan dok.
Ya, baik, terima kasih. Terkait dengan sampel yang berasal dari urin, tergantung pada desain penelitian kita. Apa yang mau kita deteksi dari urin? Kalau yang kita deteksi adalah misalnya suatu protein tertentu yang dikeluarkan oleh urin, kita tahu pada urin yang normal protein itu tidak diekspresikan ya. Jadi kalau kita mau mendeteksi satu protein tertentu, biasanya dia sudah terfiltrasi pada glomerulus dan sebagainya. Jadi harus kita pastikan dulu apakah target kita itu memang benar diekspresikan pada urin. Kalau memang benar diekspresikan pada urin, maka kita bisa melakukan pada sampel tersebut. Tapi kalau ternyata dia adanya pada plasma atau pada jaringan tertentu, maka kita tidak akan bisa deteksi pada urin. Jadi kita harus pahami dulu, pelajari dulu protein of interest kita itu bagaimana, apakah dia terekspresikan pada urin. Demikian Pak Yahya, terima kasih.
Ya. Baik dok, terima kasih atas jawabannya dok. Sangat menarik Dr. Febriana dan juga Dr. Leng. It's very interesting. There are so many questions that our participant ask. Dan saya yakin Bapak Ibu sekalian pasti masih memiliki banyak pertanyaan mengenai Western blotting. Bapak Ibu sekalian nanti bisa mengajukan kembali melalui bisa dengan menghubungi kami langsung Biosm, dan juga kami ingatkan kembali Bapak Ibu sekalian untuk bisa mengisi form feedback yang kami sediakan, yang nantinya akan digunakan sebagai daftar hadir dan juga bahan untuk pembuatan e-sertifikatnya. Baik, di sini saya sudah tidak melihat ada pertanyaan lagi. Ya, bagi Bapak Ibu sekalian, jika yang ingin bertanya langsung bisa menggunakan fitur raise hand. So ya, jadi bisa ditujukan langsung kepada siapa, nanti akan kami buka akses untuk memberikan pertanyaannya. Kami akan berikan waktu 1 menit, 2 menit kepada para peserta untuk mengajukan kembali pertanyaan.
Hi, Danny. I just see a question that maybe I can expand on it from Michael. I think it's from Michael. Right.
Michael.
Yeah, where Michael asked about in-cell Eastern versus in-cell Western. Yeah.
Okay. Yes, please.
Yeah. So, because I think Nicole has been rendering the in-cell Western, so much like Western blot, but this is not on the blot. This is staining the protein expression, protein on the cell instead. So it's much like how we do IHC, and so in-cell Western will be staining the protein on the cell instead. So it's just like you do 96 full plate, then you're staining the antibody, mostly for fluorescence because you want multiplexing, because we need to go for penetration and everything. So chemistry not really works here. Yeah. In-cell Eastern, I believe it arises from the Eastern blot, but it's mostly talking about detecting the post-translational modification, either lipid or anything else. So it will be the same thing, but it will be like how you do in in-cell Western. You will be doing it on the cell itself, but now you're detecting on the cell the post-translational modification on the proteins of interest. Yeah. So this is pretty much the same thing, but it's just one detecting protein expression, another one detecting post-translation modification. Yeah, that's it. I hope I answered your question, Michael. Thank you for your question.
Okay, thank you so much.
No problem.
Terkaleng, ya. Jadi Bapak Ibu sekalian, dari Q&A box dan juga chat sudah kami jawab dari keseluruhan pertanyaan yang ada. Jika Bapak Ibu sekalian masih ada pertanyaan nantinya, bisa langsung menghubungi kami Biosm ataupun langsung ke saya juga bisa, Yahya Ramadhani sebagai produk spesialis untuk protein teknologi di Biosm. Dengan senang hati akan kami jawab pertanyaan Bapak Ibu sekalian, baik mengenai Western blotting dan juga penerapan aplikasi lainnya. Baik, jika tidak ada yang ingin ditanyakan lagi, kami kembali ingatkan untuk Bapak Ibu sekalian untuk mengisi form feedback yang kami sediakan. Terima kasih kembali kami sampaikan kepada Bapak Ibu sekalian untuk senantiasa mengikuti webinar hingga selesai. Dan kami harap kehadiran Bapak Ibu sekalian pada webinar-webinar kami berikutnya. And also thank you for your attendance and your willingness as speaker for today's webinar, Dr. Leng and Dr. Febriana. We're grateful to your interesting and valuable presentation. Hopefully what we got from this webinar can be applied in the future. Baik, sekali lagi Bapak Ibu sekalian saya sampaikan terima kasih dan sampai jumpa pada webinar kami berikutnya. Selamat sore, salam sehat, terima kasih semuanya. Thank you everyone. Thank you, Dr. Leng. Thank you, Dr. Febriana. Thank you, Nicole.
Speaker, we take a photo first.
Yes.
Only.
Thank you, Dr. Febriana. The presentation is very, very good.
Yeah. Okay, ready for the picture. 1, 2, 3.
Terima kasih banyak Dr. Febriana atas sajiannya, paparannya yang sangat bagus sekali ya, menunjukkan bagaimana aplikasi Western blot itu sangat bermanfaat dan berguna untuk research biomedis.
Terima kasih, sama-sama Bu Lianita.
Thanks a lot. Thank you very much, Leng, for your technical information about the Western blot. Is it very useful for everyone who will do a research in Western blot? Thank you also, Nicole, for your support on this webinar.
You're welcome. Great job, Biosm team. Thanks, Dr. Febriana.
Yeah.
Thanks, Leng. Great job.
Thank you.
Thank you, everyone.
Selamat
Bye-bye.
siang.
Thanks to participants.
Ya,
Selamat
siang. Salam
See you
sehat
soon.
selalu.
Terima kasih.
Terima kasih.
Terima kasih.
Terima kasih.
Bye, everyone.
Bye-bye.
Bye.